Our lesson for today is the molecular basis of inheritance. The nucleic acids, that is the DNA and the RNA, these are the building blocks of the genetic material. And in most of the organisms, we find that DNA is the genetic material. And in some viruses, RNA is the genetic material. And what is the function of the RNA? They mostly function as messengers. Now we will know the structure of the polynucleotide chain. Nucleotide is the basic unit of polynucleotide chain of DNA or RNA, and each nucleotide is composed of three components. What are the three components? A nitrogen base, pentose sugar, that is ribose in case of RNA and deoxyribose in case of DNA, and a phosphate group. These are the three components of each nucleotide. Now, what is nitrogenous base? Basically, nitrogenous base is of two types. One is purine, another is the pyrimidine. Purine consists of adenine and guanine, and pyrimidine consists of cytosine and thymine. And uracil is only present in RNA instead of thymine. Okay. And a nitrogenous base is attached to the pentose sugar by an N-glycosidic linkage to form a nucleoside. When a phosphate group is attached to 5-OH of a nucleoside through phosphodiester linkage, a nucleotide will be formed, and two nucleotides are joined through 3-5-phosphodiester linkage, and the dinucleotide is formed. So what we understand, when numerous nucleotides are joined together, a phosphonucleotide chain is formed. We can say like that way. Understood? And one end of this polynucleotide chain, it will contain pentose sugar with free OH at 5' end. You can see in the diagram, and it is called the 5' end. And the other end, it contains sugar with free OH at 3' end. And sugar and the phosphate will constitute the backbone of the polynucleotide chain, and nitrogenous bases will be linked to sugar moiety, which will project from the backbone. Understood? I think you know this very well. Now, in RNA, each nucleotide will have an additional OH group, and more nucleotides, when they are linked together, it will form polynucleotide. Now, we will come to the structure of DNA. DNA, is made up of two polynucleotide chain, which is coiled in a right-hand fashion. James Watson and Francis Crick in 1953, they proposed this double helical model of DNA, which was based on the X-ray diffraction data produced by Wilkins, Franklin, and Chargaff. That we will come later on as the Chargaff's rule. Okay, so the backbone of the DNA will be formed of sugar and the phosphate, and they will have anti-parallel polarity. And these bases in the two strands will be paired through H bond, which will form the base pairs. A will form T and G will be forming as C. We will come in details when we go through the chapter. So this is leveling type of structure given. You can see in this diagram, it is A plus G equal to T plus C, and A plus G by T plus C equal to 1. This we will come to derivation. Now, length of DNA, how you will find out? The length of the DNA will be equal to the number of base pairs multiplied by distance between the two adjacent base pairs. Okay, suppose if you come to human beings, the number of base pairs is 6.6 into 10 to the power 10. You can see here, and the length of the data, it will be 6.6 into 10 to the power 9 multiplied by 0.34 into 10 to the power minus 9. Why? Because it is based completely on the formula. The length of the DNA will be equal to the number of the base pairs multiplied by the distance between the two adjacent base pairs. So if you solve out this, you can find out the number of base pairs. Now we will come to the packaging of DNA. The packaging of the DNA, first we will know about the prokaryotes. In prokaryotes, what happens? A well-defined nucleus is absent. So DNA will be present in a region which is called nucleoid. The negatively charged DNA is coiled up with some positively charged non-histone basic proteins, and DNA in nucleoid is organized in large loops which is held by proteins. Now, how this packaging is done basically, Roger Kornberg reported that the chromosome is made up of DNA and protein, and this another scientist came forward later on, Biddle and Tatum. They reported that chromatin fibers will look like beads on the string, but the beads are repeated units of the protein. And these proteins will be associated with DNA and are of two types. One is the basic protein, which consists of histone and protamine, and acidic non-histone chromosomal, NHC we say in short, this type of protein. Okay, and a negatively charged DNA molecule will wrap around the positively charged histone protein to form a structure which is known as nucleosome. And this nucleosome core will be made up of four types of histone protein: H2A, H2B, H3 and H4, which will be occurring in pairs. Now here 200 base pairs of DNA helix will wrap around the nucleosome and which will be plugged by H1 histone protein. Clear? In this way, the repeating units of nucleosome will form the chromatin in nucleus, which is a thread-like structure. And again, supercoiling will form loop structure, which is known as the chromatin fibers. And these chromatin fibers will further coil up and condense at metaphase stage of cell division, and they will form chromosome. Now, euchromatin and heterochromatin, these two types of chromatin fibers are found. Euchromatin are the regions of chromatin which are loosely packed during interphase. Okay, and heterochromatin are regions of chromatin which are densely packed during cell division, and these are known as heterochromatin. And this euchromatin and heterochromatin, you have to find out the difference also. And this euchromatin, they do not contain repetitive DNA sequence, and heterochromatin, they contain inactive genes. And euchromatin will contain the active genes. The heterochromatin are enriched with highly repetitive tandemly arranged DNA sequences. Euchromatin is transcriptionally active, whereas heterochromatin is inactive. Okay, you have to find the differences between euchromatin and heterochromatin. Next, the search for genetic material. The search for genetic material, here we will know about the Griffith experiment. Okay, Frederick Griffith, he conducted experiment with Streptococcus pneumoniae bacterium which caused the disease pneumonia, and he observed the two strains of this bacterium, one forming the smooth shiny colonies which is known as the S type with capsule, and another forming rough colonies R type without capsule. What happened? What he did, when live S type cells were injected into mice, they died due to pneumonia. And when live R-type cells were injected into mice, they were seen to be survived. When heat-killed S-type of cells were injected into mice, they survived and there were no symptoms of pneumonia. Okay, and after that, it was concluded that the heat-killed S-type bacteria, they caused a transformation of the R-type of the bacteria into S-type bacteria. But Griffith was not able to understand the cause of the bacterial transformation. Okay, this is the Griffith's experiment, what he did. Now, biochemical characteristics of transforming principles. Oswald Avery, Colin MacLeod and Maclean McCarthy, what they did, they repeated this Griffith's experiment in an in vitro system in order to determine the biochemical nature of the transforming principles. They reported that the DNA from the heat-killed S-type of bacteria, they caused the transformation of non-virulent R-type bacteria into virulent S-type bacteria. They also discovered that protease and RNase did not affect the transformation, while DNase inhibited this process. And they concluded now that DNA is the hereditary material. Okay, this is about the Hershey and Chase experiment. This is the blender experiment. I'm coming to this later on. Just this Griffith experiment first. Now, proof of DNA as the genetic material. Hershey and Chase, they concluded. They conducted experiments on bacteriophage to prove that DNA is the genetic material. Okay, this preparation they made, these were used separately to infect the E. coli or Escherichia coli. Here, what happened? Some bacteriophage virus were grown on a medium that contained radioactive phosphorus P32 and some in another medium with radioactive sulfur S35. The viruses, they were grown in the presence of radioactive phosphorus P32 contained radioactive DNA, and similar viruses were grown in the presence of radioactive sulfur S35 which contained radioactive protein. Okay, and both these radioactive virus types were allowed to infect the E. coli separately. Soon after infection, the bacterial cells were generally agitated to blender to remove the viral coats from the bacteria. And this culture was also centrifuged, or centrifugation process was done to separate the viral particle from the bacterial cell. Now here, after this experiment, the observation and conclusion came as only the radioactive P32 were found to be associated with the bacterial cell, whereas the radioactive S35 was only found in surrounding medium and not in the bacterial cell. And this indicated that only DNA and not protein coat entered the bacterial cell. And this proved that genetic material is the DNA, which is passed from virus to bacteria and not protein. Clear? Now, what are the properties of a genetic material, or what are the characteristics of a genetic material? The characteristics, first, it should be able to generate its own replica by the process of replication. It should be chemically and structurally stable. It should be able to undergo slow and gradual changes called mutations which result in evolution. It should be able to store the genetic information which is inhibited or inherited, and it should be able to express itself from the Mendelian characters. Okay, so these are the characteristics of the genetic material. Now the reasons for stability and reasons for mutability. Reasons for stability of DNA is due to less reactivity, and reason for mutability of RNA because it is highly reactive. So this is, you can see here the properties of the genetic material in both the cases. RNA is very unstable, so RNA virus can mutate and evolve very faster. And DNA strands are complementary. On hitting, they separate. In appropriate conditions, they come together. From this Griffith's experiment, some properties of the DNA of the bacteria, heated bacteria, did not destroy, and it indicated the stability of the DNA. Understood? So DNA to RNA to protein. So this process, DNA to RNA, is transcription, and RNA to protein, it's known as translation. Now RNA, it was the first genetic material. You know it very well. The ribonucleic acid was the first genetic material, and RNA behaved as a catalyst as well as also a genetic material. RNA can synthesize DNA, which the process is known as reverse transcription, and it's estimated that our DNA has evolved from RNA with a chemical modification. And the backbone of the RNA is made up of pentose, ribose, sugar and phosphate, and 2'OH group of the ribose will make RNA labile and easily degradable. Clear? Two types of nitrogenous bases are present in RNA. One is purine, another is the pyrimidine. Purine consists of adenine and guanine, and pyrimidines consist of cytosine and uracil. Ribose sugar plus nitrogenous base, what will be formed? Ribonucleoside. And ribonucleoside plus phosphate group, it will form ribonucleotide. Clear? Now we will come to central dogma of molecular biology, which was proposed by Francis Crick. We say as the Crick model, Francis Crick model. Now what it states? DNA to RNA to protein. This I told you right now. The transformation DNA forms RNA by the process of transcription, and RNA to protein is formed by the process of translation. Now DNA replication. DNA replication is basically the copying of the parental DNA. Watson and Crick in 1953 they proposed a scheme that DNA replication was semi-conservative. According to this scheme, the two parental strands will separate and each strand will act as a template for synthesizing a complementary strand over it. And after completion of the replications, each DNA has one parental strand and one newly synthesized strand. Now, here an experimental proof for semi-conservative model of DNA was performed on E. coli to prove that DNA replication is semi-conservative. Matthew Meselson and Franklin Stahl in 1958, they made experiments. Okay, they grew E. coli in a medium containing ammonium chloride in which N was 15 is the heavy isotope of nitrogen for many generations. As a result, what happened? The N15 they got incorporated into newly synthesized DNA, and this heavy DNA can be differentiated from the normal DNA by process of centrifugation in cesium chloride density gradient. Then what they did, they transferred the cells into a medium with normal ammonium chloride and took the samples at various definite time intervals in the when the cells got multiplied. The extracted DNAs were then centrifuged and measured to get their densities. The DNA extract from the culture after one generation of transfer from the N15 medium to N14 medium. After each 20 minutes, the E. coli was found to divide independently after each 20 minutes, and it shows an intermediate hybrid density. Okay, and this similar experiment was also found performed by Taylor, and it also replicated DNA in chromosome is also replicated was semi-conservative. Now DNA replication process, it begins at a unique and fixed point known as origin of replication or ORI. Okay, and this DNA replication is complementary strand of DNA. These double helical structure are separated by two enzymes, DNA guidase and DNA helicase, and this is called unwinding of double-stranded DNA. And the separated DNA tend to rewind. Therefore, this process is known as the initiation process. Unwinding of the double-stranded DNA, it formed a Y-shaped Y structure configuration in the DNA duplex, and this is known as the replication fork. Okay, and the other strand is formed as small stretches known as Okazaki fragments in the 5 to 3 dash direction, and during replication one strand is formed as a continuous stretch in 5 dash 3 dash direction, which is known as the continuous synthesis process, and this is known as the leading strand of DNA. Okay, and another is the lagging strand, and the Okazaki fragments will be joined together to form a new strand by an enzyme which is known as DNA ligase, and this new strand is called the lagging strand. E. coli, it will complete the replication within 18 minutes to 20 minutes, and in eukaryotes the replication of DNA will take place at 5 phase of the cell cycle, that is the 5th phase, and the failure in the cell division after the DNA replication will result in polyploidy. Clear? Now we will come to the process of transcription. Transcription, the process of copying genetic information from one strand of the DNA into RNA, is termed as transcription. This principle is complementarily it governs the process except the adenosine. However, the base pairs with uracil instead of thymine as in replication. Okay, and now we will have another new term, that is the transcription unit, the unit of transcription. The transcription unit of DNA, it contains 3 regions in the DNA. One is the promoter, another is the structural gene, and third is the terminator. Now the promoter, what is that? It is the binding site for RNA polymerase for initiation of transcription. And the structural gene, it codes for enzymes or proteins for structural function. And terminator, it is the region where transcription ends. The DNA dependent RNA polymerase will help in the DNA replication by catalyzing the polymerization in only one direction, that is 5'3' direction. Okay, and the DNA strand that has the polarity of 3'5' act as the template, and it is also known as the template strand. Okay, and you can see here the promoter, structural gene, and the terminator. Clear? Promoter is the binding site, structural gene is the region between the promoter and the terminator where the transcription will take place, and terminator is the site where transcription will stop towards the 3' downstream to 3' direction. Okay, and the strand which does not get transcripted is known as the coding strand. And the promoter will be located at the 5' end of the structural gene of coding strand and provides the binding site for RNA polymerase. And the sequence of the DNA located towards the 3' end downstream of the coding strand where the process of transcription would stop is known as the terminator. Okay, I think you have understood. Just go through it. And the initiation factor present in the RNA polymerase initiates the RNA synthesis. RNA chain is synthesized in 5'3' direction, and in this process the activated ribonucleoside triphosphate is added. This complementary to the base sequence. This is the process of elongation. An enzyme called primase will initiate replication of the stranded, which is oriented in the 3' direction, and this will generate 10 to 60 nucleotides along the primer RNA. And the free 3'OH of this RNA primer will provide the initiation point for DNA polymerase for sequential addition of deoxyribonucleotide. And DNA polymerase will progressively add deoxyribonucleotide to the free 3' end of the growing polynucleotide chain so that the replication of the 3'5' strand of the DNA molecule is continuous. And termination, the termination factor or Rho factor, it will bind to the DNA polymerase and will terminate the process of transcription. Clear? The Rho factor we say. Promoter, coding strand, and this is the terminator. In bacteria, what happens? The transcription and the translation process cannot be copied. Why? Because the mRNA will require no processing to become active, and transcription and translation takes place in same compartment. No separation of cytosol and nucleus. And 3 RNA polymerases are there, and the primary transcript will contain exon and intron, and these are non-functional, so introns must be removed. And in this transcription, in eukaryotes, here we are talking about, the structural gene are monocystronic in eukaryotes, and the process of transcription will be similar. So, will be similar to that in prokaryotes, and it will take place in the nucleus. And coding gene sequence called exon, they will form mRNA, and non-coding sequence called introns, they are removed during RNA splicing. In eukaryotes, what happens? 3 types of RNA polymerase are found in the nucleus. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Okay, RNA polymerase 1, what they will do? They will transcribe the rRNAs. RNA polymerase 2 will transcribe the precursor of mRNA. And RNA polymerase 3 will transcribe the tRNA, 5S RNA, and snRNAs. snRNA is the smaller nuclear RNAs. Okay, and there will be the post transcriptional modifications also. And the post transcription modifications, what happens here? The primary transcript which are formed, they are non-functional, and they will contain both the coding regions called exon and non-coding regions called intron in RNA, and these are called heterogeneous RNA or hnRNA. And the hnRNA undergoes 2 additional processes called capping and tailing. And in capping, the unusual nucleotide methyl guanosine triphosphate is added to the 5' end of the hnRNA. And in tailing, the adenylate residues are added at the 3' end, and this process is known as splicing. Okay, this hnRNA will undergo a process where the introns are removed and exons are joined to form mRNA. This process is known as splicing. Clear? Now we will come to genetic code. What is genetic code? The relationship between the sequence of nucleotides on mRNA and the sequence of amino acids in a polypeptide is called the genetic code. Okay, different scientists came forward and they determined the genetic code in different aspects. And the sequence of the 3 base pairs determine a single amino acid is called the codon. 20 types of amino acids are there, which is involved in the process of translation. You can see here the names of the 20 types of amino acids are there. It's given. Okay, now what are the codons used for various amino acids? You can see U, C, A, G. Okay, this is the first position, and in the middle you can see this is the second position. At the last U, C, A, G, this is given. This is the third position we say. Okay, now here what happens, you can see U, it will be converted to, this is codes given here which I told here. These codes are given here in the bracket, you can see, and these codes are used up here also in this chart. The full form of these codes are given here, which is written in yellow, so you can understand from here what it will be converted. A will form A, T, and G will be converted to C. Now codon is triplet. Triplet means 3-letter code, and 61 codons code for amino acid and 3 codons do not code for any amino acid. These are known as the stop codons. They act as stop codons or nonsense codons or termination codons. AUG, they has dual function and it code for methionine and they act as initiator codon. Okay, and in eukaryotes methionine is the first amino acid and formyl methionine in prokaryotes. An amino acid is coded by many codons and such codons are known as degenerate codons. Now what are the salient features of a genetic code? The codons are triplet. Out of 64 codons, 61 code for 20 amino acid and 3 codons rather do not code for any amino acid, and 1 codon code for one particular amino acid, hence the code is unambiguous and specific. Some amino acids are coded for more than 1 codon, so the code is degenerate. The codon is read on mRNA in a contiguous fashion. So that contiguous fashion means without punctuation. So this code is commaless. The genetic code is nearly universal. The genetic code is universal, means a particular codon will code for the same amino acid in all organisms except in mitochondria and few protozoa. AUG is a dual function codon and it will code for methionine and it also acts as initiator codon. Understood? So I have to give some salient features of genetic code. Now mutation of genetic code. How this mutation is taken place? This mutation is defined. First you have to know how it is defined. It's defined as the sudden inheritable change in the genetic material. It can be of two major types basically. One is the point mutation, another is the frame shift mutation. What is point mutation? Point mutation is the mutation in a single base pair which is replaced by another base pair. For example, if you see in case of sickle cell anemia, what happens? Point mutation in beta globin chain will result in the change of glutamate to valine. Okay, this is an example of point mutation. Second is the frame shift mutation. What is that? It is the change in the reading frame because of insertion or deletion of base pairs. Now insertion means it is the addition. It is the addition of one or more nucleotides in the DNA segment. Insertion of 3 or its multiple bases do not change the reading frame but will add a new amino acid. Okay, and deletion means the removal of one or more nucleotides from the DNA segment, and deletion of 3 or its multiple bases do not change the reading frame but will basically remove one or more amino acid. Types of RNA, mRNA, tRNA. mRNA, what is that? mRNA is the what it does basically, messenger RNA. It will provide the template for the process of translation. Translation is means the protein synthesis. Okay, and second is the rRNA, that is ribosomal RNA. Okay, ribosomal RNA structural and catalytic role during the process of translation. tRNA, transfer RNA, or we can say another is there sRNA, that is known as soluble RNA. This both brings the amino acid for protein synthesis and will read the genetic code. Clear? These are the two types of RNA. Now we will know about the process of translation. What is translation? Translation is the process of synthesis of protein from mRNA with the help of ribosome. A translational unit in mRNA, it from 5' to 3', it will comprise of a start codon, region coding for a polypeptide. A stop codon will be there and untranslated regions will be present. Untranslated region we say as UTR, untranslated region, at the both 5' 3 and end, 5' and 3' end. Three stages of the process of translation or stages of protein synthesis are there. First is charging of the tRNA, second is the initiation, elongation, termination. First, charging of RNA means the process should be started. Initiation, what happens here? First, we'll know about the charging of tRNA, the formation of the peptide bond. They will need energy, which is obtained from the ATP. And for this, amino acids are activated. Amino acid plus ATP, they will be linked to their cognate tRNA in the presence of amino acid tRNA synthesis. So, this tRNA becomes charged. This is the first. Now comes initiation. Initiation is the assembly of the ribosome on an mRNA, and activation of amino acid and its delivery to tRNA. First process, that is initiation. Next is elongation. Repeated cycle of amino acid delivery will be taking place during this process, and peptide bond formation and movement along the mRNA is known as translocation. This process will be taking part under this step of elongation. Next is termination, and this is basically the release of the polypeptide chain. Okay, this is the release of the polypeptide chain. Now I will tell in details about each. The initiation, what happens here? In prokaryotes, the initiation requires a very large and small ribosome subunits. The mRNA initiation tRNA and three initiation factors are there, which we say as IF. The activation of amino acid first takes place here, that is, the amino acid become activated by binding with the amino acid tRNA synthesis enzyme in the presence of ATP. And transfer of the amino acid to tRNA also takes place here in this process of initiation. Okay, the cap region of mRNA will bind to the smaller subunit of the ribosome. The ribosome, it has two sites, A site and P site, and the smaller subunit will first bind to the initiator mRNA, and then they will bind to the larger subunit. In this process of elongation, this is another charged amino acid tRNA complex will bind to the A site of the ribosome at the second phase or the second codon, and a peptide bond will be formed between carboxyl group of amino acid at P site and amino group of amino acid at the A site by the enzyme peptidal transferase. You have to know which enzyme helps in this process. In all the processes what is there in this chapter, you have to know about the type of the enzyme which is taking part or which is catalyzing this process. And the ribosome will slide over the mRNA to form codon to codon in the 5' 3' end. And according to this sequence of codon, amino acids will be attached to one another by peptide bond and a polypeptide chain will be formed. And this process of termination, when the A site of ribosome will reach a terminating codon which do not code for any amino acid, no charged tRNA will bind to the A site. And dissociation of polypeptide from ribosome will take place, which is catalyzed by a release factor. And there are three termination codon: UGA, UAG, UAA. Okay. Now in this process, in these steps, the process of translation takes place. Now regulation of gene expression. Regulation of gene expression means controlling the amount of time of formation of gene products according to the requirement of the cell. In eukaryotes, gene regulation can take place at 4 levels. First is transcription level, which is regulation of the primary transcript formation. Second is the processing level, regulation of splicing. Third is the transport of mRNA from nucleus to the cytoplasm. And fourth is the translation level. Okay, so isolation is done. This is the first step. Number two is digestion of DNA. Now, by this process of polymerase chain reaction, many copies are produced, and this step is known as amplification. And DNA is cut into very small fragments by treating with restriction endonuclease. And DNA fragments are separated by this agarose gel electrophoresis method. And the separated DNA fragments are visualized under ultraviolet radiation after applying the suitable dye. Okay, and this DNA is transformed from electrophoresis plate to a nitrocellulose or nylon membrane sheet. This is known as southern blotting, B-L-O-T-T-I-N-G, blotting. And VNTR probes are now added, which bind to specific nucleotide sequence that are complementary to them. This is known as hybridization. And these hybridized DNA fragments are detected by autoradiography, and they are observed as dark bands on the X-ray film. Okay, this is all about fingerprinting, DNA fingerprinting. And you can see here the steps which I said, these are given in points. Okay. Next is DNA fingerprinting applications. The applications, it is used as a tool in forensic test to identify the criminals, and it is used to settle paternity disputes. It is used to determine population and genetic diversities, to study evolution. So many points you can see here also. The forensic tool to solve paternity for the diagnosis of genetic diseases, to determine phylogenetic status of animals, determine population and genetic diversity, to genetic diseases. Okay, this is just a repetition of this here given. Okay, so these are the applications of DNA fingerprinting. This is all about the chapter. Go through it properly. Thank you so much.